Hello and welcome to the final live session of JuliaCon 2022. It's been an exciting couple of days. My name is Valentin Shuravi. I'm one of the general chairs of the conference, and it is my privilege to introduce our last keynote speaker, Husain Attarwala. Husain is a clinical pharmacologist. He's a and researcher in drug development. Currently, he's serving as head of clinical pharmacology and pharmacometrics at Moderna, and his research focuses on developing predictive models to guide clinical dose decisions for mRNA vaccines and therapeutics. And Husain, at this, I hand it over to you.
Thanks. Thank you to all the organizers for inviting me here for this talk. It's a pleasure to be here and share some of the really cool things that we're doing here at Moderna to support development of our vaccines and therapeutics using modeling and simulation. So these are some forward-looking statements that we are going to make and disclaimers. So jumping in, so what my role here is at Moderna is that I head up the clinical pharmacology and pharmacometrics department here. So we do model-informed drug development using modeling and simulation. So what is pharmacometrics? For those who are not familiar, pharmacometrics is the science of interpreting and describing pharmacology in a quantitative fashion. So by pharmacology, we mainly look into pharmacokinetics and pharmacodynamics. Pharmacokinetics is the mathematical understanding of rate of absorption, distribution, metabolism, elimination of the drug and its metabolites. In simple words, what body does to the drug is PK. Pharmacodynamics, as you will hear more and more in the talk, is the study of kinetics of pharmacological response at different drug dose or concentration. In other words, once the drug is in the body, what are the effects of the drug on the body, and how long do they last? What is the relationship between dose, concentration, etc. is pharmacodynamics. Uh, looking at it visually, so once a drug is administered, it gets absorbed, distributed, metabolized, and eliminated from the body. When we collect blood samples at various time points, we see that concentrations rise initially as it is absorbed, reaches a peak, and then disappears from circulation over time as it is distributed or eliminated. The concentration at various sites in the body leads to effect and the relationship between the effect and concentration is called pharmacodynamics. Now, once we combine PK and PD, we get a PK-PD relationship where you can see effect versus time, and the mathematical understanding of these kinetics and dynamics over time course is what we describe using different models in the using ordinal differential equations and whatnot in pharmacometrics. Yeah. So why is it important? So pharmacometrics plays an important role at various stages of drug development. During early phases, modeling and simulation helps in guiding dose selection for first-in-human studies. In phase 1 clinical study, pharmacometrics helps in understanding dose response, and exposure safety relationships, which helps in predicting dose levels for later phase studies. At the end of phase 1 or phase 2 studies, modeling and simulation plays a very important role in selection of phase 3 dose. Uh, it is important as safety and efficacy are linked to dose and exposure. And as you can imagine, if the dose is too high, it may show unintended adverse events. And if it is too low, it can lead to suboptimal efficacy. And indeed, selection of the wrong dose is one of the very topmost reason of why many drugs fail in phase 3 studies. Using modeling and simulation, we really get help in predicting safety and efficacy outcomes using prior data while going through various learn, confirm, and predict cycles. Uh, So once we have selected a phase 3 dose, we have conducted a phase 3 trial. Then at the end of phase 3, again modeling and simulation plays a very important role in understanding covariate effects, dose, exposure, safety relationships, and what kind of dose and um, needs to be taken for different subpopulations, and that ends up on the drug label, which is uh, on the drug label, approved drug label. The dose that you see is backed by a lot of actual data and the modeling and simulation that has been done to support it. So, in our talk today, I will walk you through three examples where we have utilized modeling and simulation to guide optimization at various stages in drug development. And before we jump into our examples, let's briefly look at the workflow and tools we use to achieve our modeling and simulation objectives. So, to achieve what we saw in the previous slide, as you can see that it span across the development life cycle of a drug from early stages, preclinical to clinic, to all the way to approval and post-approval. Uh, in general, there are a lot of different tools that are available and used to do this kind of analysis. So those include like NCA, non-compartmental analysis, population PK/PD analysis, PBPK models, QSP models. Um, I will not go through in detail of what all of these are, but to enable dynamic drug development, and go through multiple cycles of model creation, validation, iterative simulations. Uh, we need tools that could enable that. And there are like variety of tools available, but there is not like one tool that can do most of it or all of it. The Puma's software within the Julia ecosystem provides a one-stop for all sorts of pharmacometric analysis and helps in accelerating model development and simulation workflows. And it was important for us to have such an innovative tool to keep up with the pace and the speed that we uh, entered into and execute those workflows. And as you can see, the statement from FDA, which says that model-informed drug development approaches are extremely important for maximizing the probability of success across different clinical studies. And there are a lot of examples where these approaches have become very useful and changed the course of drug development into success. Yeah. So to have a tool that is acceptable by regulatory agencies, uh, those tools needs to be robust, validated, reproducible, and need to deliver results in time such that we can make those decisions when the need is to be made. So after rigorous testing and validation, we engaged Pumas to tailor all our modeling and simulation needs for the MIDT program, and we use it here at Moderna extensively. So, going through now the examples, now let's jump into the case studies. Uh, so first, we will talk about MIDD for therapeutics, where I will discuss how we used it for first-in-human dose selection, and then we will go into MIDD for vaccines, where we will showcase how we have been using modeling and simulation for our vaccines phase 3 dose selection, and then dose selection for pediatric populations. So, going into the therapeutic side first, here I will show walk through the example where we had a lipid nanoparticle which encoded heavy and light chain of an antibody. So the lipid nanoparticle with mRNA encoding heavy and light chain of an antibody is administered in vivo to animals, and then this mRNA expresses these functional proteins, and a functional antibody is secreted out of the cell by using mRNA technology. So at our end, when we were developing this model, when we only had non-clinical data, the objective was to understand rat to human translation of PK and PD responses, and then to predict what the human response would be based on the translational understanding that we gained while modeling those outcomes. And these are some of the data that we had. So we had data across various dose levels in RAC and NHP after IV administration, and we had PK and PD readouts measured over time. So what we know from small molecules in antibodies is uh, that as we go in higher species, the different rates of metabolism, of absorption, elimination, they slow down. Why? Because if you look at heart rate in rats, for example, it's much faster than in humans. Blood flow rates are much faster than in humans. So overall, small animals are the systems are much faster than what we have in humans, and we see that resulting concentration time profiles that show that the elimination profiles are slower as we go up in humans. So this is something called principle of allometric scaling, that as we go up in species, the time course of response lasts longer due to that slowed metabolism and overall system. This principle works very well for small molecules and antibodies. In most cases, there are exceptions, but for our case, we wanted to see if this applies to mRNA PK as well. So we embarked on the journey and used our data that were available to build a PK/PD model. So here, this is a model schematic where we model it using compartmental models. Each compartment is an ordinal differential equation, and the rates and to and fro, to and from different compartments are modeled and parameterized using several understanding of physiology and some of the parameter bounds that are plausible. So top portion is a PK model. Once there is drug concentration, then we link it to the PD response. So the IgG is the PD response that we had with this mRNA 1944, and that is also linked via this PK/PD modeling framework. So now let's look into the data. So here, uh, what is shown is plasma mRNA PK profiles over time in rat and NHPs. And as you can see, the half-lives in rat and NHPs are different, where rat we have a shorter half-life, NHPs we have a longer half-life. And when we apply the principle of allometric scaling in the modeling framework, we can fit the data across both species by uh, applying those exponents of clearance and rates. So body weight based allometric scaling described the mRNA PK well in rats and monkeys. Then we looked into PD, which for antibodies it is well known that allometric scaling works quite well, and in this case also it worked beautifully, where uh, the observed data fitted quite well when this principle was applied. So once we have a translational model and we can describe some of the interspecies differences using some of the known principles that we have, we can then use that model to predict what human response would be. So we had at the time, the question was what dose should we select, what dose range should be taken in the human clinical studies, and whether that dose is going to lead to concentrations that are deemed to be effective in humans. So we used this translational approach, extrapolated to humans using the same allometric principle, and predicted what the outcomes would be in humans. So here, this is the prediction across three different dose levels: zero point 0.1, zero point 0.3, and zero point 0.6. The solid line is the median prediction, and the shaded area is the prediction interval. And based on this, and there were many more other simulations done, and these are the doses that we ended up selecting based on the model simulations. Um, and 1 microgram per ml was the threshold that we were targeting, and we wanted to see that those concentration last over a certain period of time. And we can see here that at zero point 0.3 mg per kilo, they would last at least up to 4 months after a single dose. Then we actually conducted the clinical trial and we got the observed data. And now let's see how the observed data overlaid with what we predicted a priori using non-clinical results. So as you can see here, observed data overlaid very well with what we had predicted at all 3 dose levels. We were very happy to see this because this was the first time ever when a mRNA based therapy was used to uh, produce antibodies within an individual. So it is our own body producing the antibodies by administering mRNA, and we got a very nice dose dependent response at these 3 dose levels. And it also was very exciting to see that how the translational framework that we had worked so well in this case. So we are using this across many other programs, and each different modality, each different target has its own different considerations that we take into account in predicting responses for humans. So in summary, administration of mRNA 1944 resulted in dose-dependent increases in levels of antibody against chickpea-24 in rats, NHPs, and humans. Body weight-based allometric scaling of PK and PD model parameters adequately describe the observed PK-PD profiles in rat and NHPs, and dose-dependent pharmacology in humans was adequately predicted from preclinical species. Now, jumping on to pharmacometrics for vaccines. So, uh, pharmacometrics for therapeutics is very widely used in biopharma industry. Um, there are many examples of stories and success, a lot of models out there, and this is something which is now an expectation in therapeutics side of things. For vaccines, this was a brand new chapter. So right now, if you pick some of the books on pharmacometrics, you will not find a chapter on vaccines. So here at Moderna, we embarked into developing these models so that we can use modeling and simulation approaches to guide dose selection for vaccines as well. Uh, uh, and especially in clinical development, dose selection setting where we could influence those decisions. So what, how we embarked on the journey. So we set out to develop pharmacometric models for our vaccines. The model describes the immune response stimulated by a vaccine and the resulted immune dynamics. So we call the model immunostimulation immunodynamic model, or ISID model for short. Uh, Now let me walk you through the model development. As with all pharmacometric model, we have to first understand the underlying mechanism of the medicine, or in this case, the vaccine. With vaccines, we know that they stimulate immune responses after each vaccination, uh, and immune responses can be measured in multiple ways. The most common and widely accepted measurement of an immune response after vaccination is measurement of the neutralizing antibodies. Here we are looking at a typical time course of neutralizing antibody response generated after vaccination. After each vaccine dose, our body generates an immune response via immune stimulation, which persists over a certain period. In developing model, we first set out to mathematically describe the underlying mechanisms of immune stimulation and dynamics of this response. So, what are these mechanisms and how do we represent them mathematically? So here, this is a simplified conceptual representation of mechanisms underlying the generation of neutralizing antibody response following vaccination. Following the first vaccine dose, naive B cells are activated to produce antigen-specific antibody-secreting plasma cells. Some of those activated B cells form memory B cells, which are amenable to activation upon second vaccine dose. After the second vaccine dose, naive B cells as well as memory B cells are activated to produce antigen-specific antibodies. These are the underlying mechanisms, but how do we represent them mathematically? Well, there are a lot of mathematical equations that were needed to describe this. We started out with simple models, and then uh, saw if the model was capturing the observed data and what else was needed to be included, what are the different functional forms that are needed to describe these responses. And these are the some. And at the end, we had a model that had 10 differential equations and 17 parameters to adequately describe the time course and stimulatory dynamic effects that were said. Now, to understand all of these differential equations, I will need to give you a walkthrough of each equation, what each parameter is, but I will spare you the pain and not go into all of those details here. Um, and I just wanted to show you that what it looks behind the scenes. Uh, so here, why develop such a model and what is the use? So the key questions that were in front of us, and there are many questions that can be answered, like what is the dose, what is the regimen, uh, what, and what are different correlated effects. But here we will focus on two main questions. So first one is what vaccine dose should be used in clinical studies, and the second, how do we select doses for different age groups? For me personally, the most important question was what vaccine doses should be given to my 6 month old son and 2 year old daughter. So it was a very personal thing, and it is incredible that we had a chance to work on something that our own children would get. So Uh, the two examples that we will go through are here. So first one, we will review the ISID and reactogenicity modeling for our CMV vaccine. Uh, and I'll walk you through that first, and then we will go into ISID modeling for our mRNA 1273 vaccine, where we used it for our pediatric dose selection. When we we began developing these models back in 2019, we had phase one clinical data readout from our CMV vaccine. So our model, the development of these models began before the pandemic, when COVID was not around. So we started way back then, and since now we have these models, we were very nicely and readily able to use it for our COVID vaccine as well. So, what is our CMV vaccine? So, our CMV vaccine includes 6 mRNAs that encode two antigens, five encode the pentamer, and six encode the GB antigen. And each antigen infects different cell types. And we have developed ISID models for both antigens. We began so we began developing data with our phase one data in hand. So here we had data for neutralizing antibodies against epithelial cell at different dose levels, 30, 90, 180, 300, at different time points measured. And then we wanted to see if we can describe this using the mathematical framework we had. And this is the data for our antibody response against the GB antigen. So we had both of these data and used this for developing our ISID model as well. So we went through hundreds of iterations to find the optimal model structure, parameters, and then qualified our ISID model for neutralizing antibody titers against epithelial cell infection based on various numerical and visual diagnostics. Our final model described the observed data well. Here the dots are observed data, and solid line is fitted mean titer levels, and shaded area is the 5th to 95th percentile interval. As you can see, the model adequately captured the central tendency of the data as well as the inter-individual variability. We did a similar exercise for our GB antigen data, and were able to qualify the model using different numerical and predictive diagnostic. So once we have a qualified model, we can use it for performing clinical trial simulations and answer some of the questions that are at hand. The key question that we had in front of us was, which dose should we select for our phase 3 clinical study? And as I previously mentioned, a selection of a phase 3 dose is one of the most important decisions during development of a drug, as it could uh, lead to maximize the success tremendously if we are good at making those decisions. So here we used our model to simulate relationship between dose and neutralizing antibody titers across a wide dose range and evaluated which dose is expected to yield neutralizing antibody titers above the benchmark of natural infection. In this plot here, dose is on x-axis and neutralizing antibodies at 1 month post third dose are on the y-axis. Solid blue line is the geometric mean titer, and shaded area is 5th to 95th percentile prediction interval. We we saw that a 50 microgram dose was enough to achieve GMT above the GMT benchmark of natural infection for both epithelial and fibroblast cell infection. We could possibly get a greater response if we went to 250 microgram, where the titer exceed the benchmark in majority of population for the neutralizing antibody against fibroblast cell infection. But dose selection is not just about efficacy. We also take into consideration safety. So, how about adverse reaction at those higher doses? So, in order to model the relationship between dose and adverse reaction, we used a typical logistic regression model. Here on the graph, we have at a given dose, an individual may either have no adverse events or they may. And we do this, we collect data across a variety of different adverse events that are there. And once that data is available, we can use it to derive a probability of such events given a certain dose. And once we have that kind of probabilistic analysis done, we can derive something called a composite risk score. Composite risk score is a combined probability of having any adverse reaction. On the slide, you can see the composite risk score on the y-axis and dose on the x-axis for each vaccine dose. Uh, Composite risk score was modeled using data from our phase 1 clinical study. Probabilistic modeling helps to produce a continuous relationship between dose and risk of adverse events. So even at doses that are not evaluated in the clinical study, you can have those continuous relationship build, and you can visualize that where are the curves bending in a way that we do not want them to bend, and then we can base our decisions where the those risks are minimal. So now, if you look at our 250 microgram dose, which gave us really high immunogenic titers, you can see that the risks are slightly higher. They are not way high, but the slightly higher risks of grade 3 reactions at 250. If we dial down the dose at 100, then the risk of those grade 3 events, which and grade 2, grade 1 also, is low. So uh, it appeared that 100 microgram would be a favorable dose from a safety standpoint. Now, if we go back to our dose response relationship from the efficacy side of things, where there is uh, where we see immunogenicity, we can see that 100 microgram dose had a lower had antibody titers that were above the benchmark of natural infection in majority of subjects. And uh, the basis of dose selection is to maximize immunogenicity while retaining minimal risk of adverse reactions. So based on both safety modeling and efficacy modeling, uh, we selected a phase 3 dose of 100 micrograms for a CMV program. This dose is now an approved dose for our phase 3 trial, and our phase 3 trial is successfully enrolling patients in this study at this dose. Uh, so we uh, did all of this modeling and then submitted all of this work at different regulatory agencies globally, including US FDA, EMA, TGA, and many other countries across the world using the Puma software. Now we are switching gears to ISID modeling for our COVID-19 vaccine. Here we will see how we successfully use this modeling approach for our pediatric dose selection. The for uh, our COVID vaccine, at this time we had a lot of data. We had data available from uh, 2567 subjects uh, from our phase 2 clinical studies in adults, adolescents, and older children, which were greater than 6 years of age, and from our phase 3 study in adults. The modeling approach allows for pooling of data across different studies and helps to derive inferences from totality of clinical data. Versus if we just look at observed data and try to make our decisions, then you're looking at one set of data at a time. Using a model, you can integrate all different studies and uh, get a holistic understanding of our responses in different age groups. And then we applied some of the scaling mechanisms that is there to predict what doses would be in our pediatric trial. So how this was done, we adapted our ISID modeling framework to develop that we developed for CMV vaccine to COVID, uh, and we optimized some of the parameters that were specific to COVID-19. Uh, we assessed dose and age as covariates on antibody titer response, and then simulated antibody titers at various doses across different age groups. So, what was our goal? Our goal was to predict which dose level in young children and infants would yield neutralizing antibody titers that are similar to young adults. On this graph, the reference immunogenicity data is from our phase 3 co study in young adults. And then we used our ISID model to simulate distribution of antibody titers at different doses. Uh, and then we use it to evaluate. We did this for a lot of different dose levels, and this was before we had our we did the actual pediatric study, the large trial, and the real question was what kind of doses will be effective. If we have a 100 microgram dose in adults, what dose is going to yield similar antibody response in uh, young population? And for vaccines, this kind of modeling is very new and done for the first time. Yeah. Uh, so once we have simulated this, we compute a metric called GMR, which is geometric mean ratio, and then there are some set criteria which are accepted by the regulators that uh, meet the statistical criteria to either pass or fail based on the results that are obtained. So using simulations, we these were the results of our simulation back then. This was done in fall of last year, um, where we simulated at different doses, 10, 25, 50, and 100. And then based on model simulations, we predicted that a primary series of 25 microgram was predicted to meet the non-inferiority criteria in young children and infants. We, as I said, it was back in August last year when observed data in these age groups were not available. The model predictions were useful in selecting doses to evaluate in the clinical study. Now we have actual data here that we got from our clinical studies. We actually conducted those trials and got our data. And I'm extremely happy to share that what we predicted back then in fall lined up with what the observed data showed when we received those data. And as you can see, the observed data closely correlates with model predictions, validating our modeling predictions and the modeling approach. and shows that how integrating the modeling and simulation workflows during the clinical development pipeline can help in sound decision making for our dose selections and can lead to maximize the rate of success. So, in summary, modeling and simulation helps drive data driven decision making for clinical dose selection. Traditional PKPD models are widely used for therapeutics, and we have adapted those traditional PKPD models for to vaccines. ISID models can be used to successfully predict neutralizing antibody responses across various age groups, and our ISID models can predict a priori immunogenic responses in various age groups. And we have demonstrated utility of these models for phase 3 dose selection for a CMV vaccine in this case, and used this successfully for a pediatric dose selection for a COVID-19 vaccine. And thank you everyone for your attention and listening to my talk. At Moderna, our pharmacometrics team is expanding. We are hiring. We have several leadership roles in different therapeutic areas. This is just the beginning for mRNA medicines and mRNA science. There's a lot more to be learned, done, and a lot more people to be helped suffering from different variety of conditions and diseases. So we have different, apart from vaccines, we have autoimmune therapeutic area. There is oncology. There are a number of programs there, and there are rare diseases. And we are also building a QSP modeling function within our pharmacometric group, metrics group. So if you or anyone you know who has the skills, please go on our website and apply for these positions. Yeah, and that's it. And many thanks to our a lot of work has been done in collaboration with our Pumas.ai team there. In particular, Vijay has been one of the key individual who we have collaborated with over several years in testing, validating, deploying the Pumas, and also consulting for clinical pharmacology and pharmacometric, leading to some of the regulatory submissions. And yeah, so many thanks to them. And yeah, that's it.
Thank you, Hussein. There are some questions. One of them is, what are the biggest challenges you're facing with software tools when you do these kinds of analyses?
Yeah. So one is the integration of different workflows, right? So in when we think about therapeutics, one minute, let me just move this here. Yeah, so different software, so for different types of analysis we have different software tools where we go into those softwares, do the analysis, then results from those analyses are then imported into other software tools that then do those analysis. And so we have to like keep jumping between different tools. That is one. It is not a challenge but a kind of a hassle. People but if something can do within a single software, then we would prefer that. uh, then other challenges, like these are software tools that are there since quite long time, and there has been some progress but not a lot of progress. So uh, like some of the time efficiencies and things like that, uh, if you see a few seconds of increase improvement in time on a small model, think about if you have a large model, and that saves you maybe 1 hour, 2 hours, and if you have to run those models hundreds of times to validate test, uh, so that is a challenge. And if we have something that is faster, then we can actually use that model to drive a decision, because decisions are timely. If we do not get our data models predictions in time, uh, then we are not able to help influence that decision, because decisions are to be made when the time is in constraint. So yeah, so that is the big one.
Thank you. Um, there was one question: do you have any specific experiences within the teams using Julia before they came convinced it was a good tool that made people go, "Okay, this thing is cool. We should use it."
So I was introduced to Julia via Pumas. So when Pumas came along, and me personally, I was intrigued by how everything else can be integrated within a single workflow, especially the simulation workflows. Some of the tools that we have, the simulation workflows are very um, time consuming, and here in Julia, it is once you have a model, it is very easy to get into those kind of things. So that is where got me intrigued. And then it was a journey to test and validate over time, and we did that in record time here. Yeah.
Okay. Moderna has been very successful in applying Julia-based software to PKPD modeling. Which other areas of drug discovery do you think could benefit from Julia being used more widely?
Um, uh, so there could be applications across all sorts of uh, modeling, right? So this is ordinal these ordinal differential equation models are used in even uh, manufacturing, like so some of the reaction systems that are there. uh, there it can be applied, but again, some of the uh, benefits and things like that will need to be weighed there, where how practical is it to jump from the current tool to a Julia tool.
Yeah.
Yeah, if because those if those models are not that complex, and the gain in efficiency, how big is it? So but I can certainly see that it can be used there. Then there are computational side. I think there are other departments already using it at Moderna. uh, yeah.
And maybe as the last question, you showed models that worked well. How often does it happen that a predictive model turns out to not be working once you acquire it and then verify the data afterwards?
So it is a continuous learn, confirm, predict cycles. So uh, it is not that one we create one model and that's it, model is set. Then at the end there is a because if we go from a very um old or model which is informed by very little data and compare that with the final, it may not correlate. So the journey is to guide at every stage. So each stage there are different questions. So for to answer a question, this is the model, this is, and we are able to at that micro stage, we are able to do quite well in guiding that. Then once we get more data, we keep on updating. So, for example, our COVID model, uh, where we had that 2,000 subject data, that was a lot of data that went into that model. So, with that kind of data, then in likely cases we are able to successful, as long as the model is good enough, tested, validated in a way that should be. Yeah. The cases where it may fail is, let's say, there is a preclinical model uh, which you base certain assumptions on, and you go into humans with certain unknowns, that how much is the receptor density, how is the um, expression of certain targets within the body that clear those moieties. And if those are off by huge amounts, then your predictions will look not very much aligned. Uh, so that is where we do all kinds of sensitivity analysis to see that, hey, uh, what kind of things, which parameters are important, and if they vary by a certain extent, then what will happen here.
Thank you. Thank you for the insightful talk, and I look forward what Moderna does next with Julia.
Thank you.